Tobacco smoke initiates cancer through a stepwise biological cascade, beginning with chemical exposure and progressing to DNA damage, mutation, and ultimately tumor formation. In this presentation, we'll follow that pathway from the first inhaled carcinogens to the irreversible genetic changes that drive malignancy. Tobacco smoke contains more than 60 proven carcinogens spanning several chemical classes, each with distinct biological behavior. These include polycyclic aromatic hydrocarbons like benzo-A-pyrene, tobacco-specific nitrosamines such as NNK and NNN, aromatic amines including 4-amino bipethyl, and volatile compounds like benzene and 1,3-butadiene. Each cigarette delivers milligram-level quantities of these agents, with aldehydes, volatile hydrocarbons, and nitrosamines contributing heavily to systemic exposure. Most of these compounds enter the body as inert pro-carcinogens. Cytochrome P450 enzymes, CYP1A1, CYP1B1, and CYP2A13 in the lung, and CYP1A2 in the liver, convert them into highly reactive electrophiles capable of binding DNA. Although these enzymes normally support detoxification, their activity paradoxically generates the ultimate carcinogenic species. Phase 2 pathways, including glutathione S transferases and UDP glucuronylyl transferases, can neutralize these intermediates, but when activation outpaces detoxification, as in individuals with high-activity CYP variants and GST-M1 or GST-T1 null genotypes, DNA damage risk rises sharply. Urinary biomarkers quantify internal exposure to these carcinogens. Total NAAL, derived from the potent lung carcinogen NNK, appears only in smokers or those with significant secondhand exposure. Levels of 1-hydroxypyrene, a marker of PAH exposure, are roughly doubled in smokers. Metabolites of benzene and 1,3-butadiene, TTMA, SPMA, and MHBMA often reach even higher concentrations, reflecting the abundance of these volatile compounds in mainstream smoke. The pivotal biological event occurs when reactive metabolites form covalent DNA adducts. Benzo-A-pyrene diol epoxide creates bulky lesions at guanine residues within TP53 mutational hotspots. NNK generates pyridinobutylated adducts and methylated bases, such as O6-methylguanine. Oxidative stress from smoke further produces ethano-DNA adducts through lipid peroxidation. These adducts serve as the physical precursors to mutation. Human tissue analyses reveal elevated adduct burdens across multiple organs in smokers. Laryngeal and oral tissues accumulate 4-amino bipethyl DNA and propionyl DG adducts. Lung tissue shows increased BPDE DNA and pyridinobutyl DNA adducts concentrated at KRAS and TP53 hotspots, detectable as a diagonal radioactive zone in 32P post-labeling assays. Bladder urothelium and cervical tissue also exhibit characteristic adducts, demonstrating systemic distribution beyond the respiratory tract. Cells deploy several repair systems to counteract this damage. O6-alkyl-guanine DNA alkylase directly reverses O6-methylguanine, but is inactivated in the process. Base excision repair, initiated by glycosylases like OGG1, removes oxidative lesions such as 8-ox-guanine. Nucleotide excision repair eliminates bulky adducts, and deficiencies in this pathway markedly increase susceptibility to tobacco-related cancers. Double-strand breaks from oxidative stress are resolved through homologous recombination or the more error-prone non-homologous end joining. When replication encounters unrepaired lesions, translesion synthesis polymerases, particularly Pol-eta and Pol-kappa, bypass the damage at the cost of accuracy. This process generates characteristic mutational signatures, G to T transversions from BPDE adducts and 8-ox-guanine, and G to A transitions from O6-methylguanine. These patterns are strongly enriched in smoking-associated lung cancers. These mutational pressures converge on key oncogenes and tumor suppressors. KRAS mutations, especially GGT to GTT transversions at codon 12, lock growth signaling pathways in an active state and are common in lung adenocarcinoma. TP53 accumulates inactivating mutations at defined hotspots, disabling apoptosis and DNA repair checkpoints, changes particularly prevalent in small cell lung cancer. Together, these alterations remove proliferative restraints and compromise genomic surveillance. Beyond point mutations, tobacco smoke induces broad chromosomal instability. Loss of heterozygosity frequently occurs at tumor suppressor loci such as 3p, FHIT, 9p, P16, and 17p, TP53. Additional manifestations include micronucleus formation, elevated sister chromatid exchange, and aneuploidy, reflecting widespread genomic disruption. Inherited polymorphisms significantly influence individual susceptibility. CYP1A1 variants like MSPI and IL-462-Val increase production of carcinogenic epoxides. The GST-M1 null genotype, present in about half the population, removes a major detoxification pathway. When high activation capacity coincides with absent detoxification, lung cancer risk rises dramatically, with odds ratios exceeding 16 even among slight smokers. Variants in DNA repair genes further modulate risk. XRCC1Q399 reduces base excision repair efficiency. OGG1C326 impairs removal of oxidative lesions and approximately doubles lung cancer risk. XPD-Q751 compromises nucleotide excision repair of bulky adducts. These variants do not cause cancer alone, but heighten vulnerability in the presence of tobacco carcinogens. Tobacco smoke acts as a systemic mutagen that co-opts metabolic pathways to generate DNA reactive species while overwhelming multiple layers of genomic defense. The combined effects of carcinogen exposure, metabolic activation, adduct formation, error-prone replication, and impaired repair create a continuous trajectory toward irreversible genetic damage. This mechanistic framework supports the classification of tobacco smoke as a complete carcinogen operating through multiple parallel pathways with no evidence of a safe exposure threshold.